Welcome, everyone. My name is Alexander Stepanovic, founder of Cascades Analytics, a media and advisory platform focused on the psychedelics and neuroplastogens pipeline. We go where the action is, speaking with the founders, scientists, and regulators shaping the next wave of psychiatric drugs. Joining me today is Dr. Deborah Mash, a true pioneer in ibogaine research. In 1993, she ran the first FDA-approved ibogaine clinical trial, co-discovered noribogaine as ibogaine's active metabolite, and spent nearly a decade conducting clinical work with opioid-dependent patients in St. Kitts. She co-founded DemeRx, and their lead candidate, DMX 1001, oral noribogaine for alcohol use disorder, just had its IND accepted by the FDA with a phase 2 trial planned for 2027. All of this is happening as ibogaine steps into the political spotlight. A presidential executive order has put it squarely on the map. The heat is on. Deborah, thank you so much for joining us today. How are you?
I'm well. Thank you for inviting me.
So why don't we dive right in? My first question for you is that 30 years from your first IND in 1993 to last week's IND acceptance for DMX 1001, what does this moment feel like?
It's been a very long journey for me, but a longer journey for the patients who need to have access to the molecules. So it was a historic moment. It is potentially a legacy action for our president. And the fact that this is the first time in my lifetime that the ibogaine research movement, the possibility of advancing the drug into clinical trials, was given wind in the sails. You know, it's been a very long, arduous, uphill, unfunded, and the list goes on from there. And then, you know, the vast underground ibogaine experiment has led to adverse events that we have to keep, you know, coming back to and trying to explain. And people, you know, if they take ibogaine in unsafe settings with impure drug product, with perhaps well-meaning, not always, practitioners who don't understand risk mitigation of this drug product, investigational drug product, then, you know, we put more hurdles up in front of us. So it was a very happy day for me on a personal, and you know, this is something, this work has always been dear to my heart since the very first patient that I saw take ibogaine for a long-standing opioid abuse problem, chemically dependent to opioids, take a single dose of ibogaine, and in 24 hours to 48 to 72 hours demonstrate an amazing transformation. And I will go to my grave with those, you know, first few patients that I saw treated in Amsterdam back in late 1992.
And you never gave up. I mean, you doubled down. You went to St. Kitts to build to lead that program. That's incredible.
I did double down, and the reason I doubled down, you know, really who motivated me more than anything was the FDA, because when I came back from Amsterdam and I went to my colleagues at the University of Miami, and I said, let me tell you what I saw. And I had a good reputation at the school, you know, the university. I was a up-and-coming faculty member. I had NIH funding. I was publishing, you know, all the good things that you're supposed to do as a faculty member. So I went to the dean of research and to other, the provost and others. And I had champions there behind me because this was risk, you know. But I also knew you know, what ACT UP had accomplished, right, for HIV/AIDS.
Mm-hmm.
And if these, you know, if we don't have, you know, sort of a movement to to get drugs to go fast. And pharma, dictates what are the blockbuster drugs. And this molecule didn't fall in that category. So we knew straight away we weren't going to get a pharmaceutical partnership. So I did what any good academic investigator would do. I went to the NIH and I said, let me tell you what I just saw. And I had a colleague who went with me because I needed to have, a backup set of eyes, right? Because they would say, that woman, But I had a colleague, MD, PhD colleague with me, Juan Sanchez Ramos, Dr. Ramos. And we both, brought back the testimonial, and then I took that to NIDA. University of Miami said, okay. They gave us a green light. And I went to the FDA, and it was amazing because the FDA was always collaborative. I mean, the doctors there, they really, you can see they're, these are people who are dedicated to the work that they do. They're government, and they're really dedicated. And my experience with them was incredibly collaborative because I had never done, and I had never submitted to a regulatory agency. I'd never done it.
Mm-hmm.
So I was on a learning curve, but I worked very hard and diligent, and I had a lot of wonderful collaborators. I can't name them all here, but I mean, believe me, I assembled what I believe to be a stellar team, and it took a team to do this. But we went in, and the FDA gave us guidance and gave us permission. And then in 1995, I went back to the FDA because Howard Lutzoff, who was continuing to treat patients, had an adverse event in the Netherlands, and that closed down any treatments going on over there. I was able to work with the mother and get the right information and of what happened in that case, presented that to the FDA, and along with some other data. FDA was happy with us, and then they gave us the full green light to go forward with an external data safety monitoring board up to what would be today psychedelic doses of ibogaine. So I got started at the University of Miami with a little bit of money that we had. And I went back to the NIH and said, will you consider, and they had site visited us in Miami. One of the deputy directors of the medication development division at NIDA came down to site visit my team at spent a day and a half with us in residence, and then went back. And basically what NIDA did then was to develop their own protocol separate from us. Yeah. And I still participated with them and shared all the information that we were getting, because we were still getting data from Howard Lutzoff. And I was collecting more human data and more human data and more human data to inform and provide support back to the FDA. But they eventually, NIDA just had a public meeting, an advice meeting, and they decided that they weren't going to fund ibogaine. Not with me, not with us at the certainly, and not with inside intramural or under NIDA's auspices, let's say.
Do you know what triggered that decision?
Well, I think there was a lot of factors. One, NIH doesn't really develop drugs. I mean, and NIDA does have the clinical trial network, and that has helped. But this was a psychedelic medicine at a time when that wasn't happening. I mean, there were only 3, to my knowledge, at that time. It was Rick Strassman with DMT, and I think he was at the University of New Mexico at the time. And he wrote a fundamental paper on how to get a DEA Schedule I license when you're, advancing a clinical trial program. And that was, we read that paper like five or six times, cover to cover, because that was guidance for us. And then Charlie Grow about it out in California with the work that he was doing. And he had done some work with MDMA and also did some work with ayahuasca at ex-US. So that was pretty much it, because Rick Doblin's group hadn't started. Michael Mithofer, Dr. Mithofer, and the group with Rick Doblin hadn't started yet. So when they had the 1995 hearing, they brought in all these, key opinion leaders and also pharma people. And basically at that time, the committee the way I read the meeting, okay, and I haven't read the minutes in a long time, but the way I recall was that the committee was split. There were some of the academics who said, yeah, this should be tested. Let's push forward. And there were others who said, no, no way. And then there were some of the pharmaceutical people who said, I think the metabolite is more interesting, which wasn't lost on me, you see. And we had discovered the metabolite. I mean, we characterized the metabolite in my laboratory with my colleagues, and worked it up in the test tube and took it into animals and measured it in human, did bioanalytical assays in urine and blood. And so we had information. And we worked up the genotyping with a brilliant head of the metabolism division at Pfizer, Dr. Scott Obach. And then Rachel Tyndale up in University of Toronto, who I had heard give a talk. I think she was, if I recall, she was presenting at the NIH, and this brilliant woman, a young woman, gets up there and is talking all about cytochrome P450s. And at the time, that was an emerging clinical, scientific and clinical area of interest. And I was in the back of the room, and I remember walking all the way up, and she was still at the podium talking to other people, and I stood there, and I talked up to her, and I said, introduced myself and said, will you genotype, or will you do a post-hoc analysis and help me with the genotyping of the cytochrome P450s in our patients taking ibogaine in the Caribbean? And so it began.
Yes, and that genotyping is actually a very good way to assess how fast ibogaine is metabolized into noribogaine, and there's a good correlation between the cardiotoxic effect and the hallucinatory effect and the metabolization of ibogaine into noribogaine. Is that right?
Yes, it is. It is. And what was interesting, so we had done low doses of ibogaine in the clinical trial at the University of Miami. And what happens when you give a low dose of ibogaine? It all gets converted to noribogaine. So in other words, if you microdose, everybody talks about microdosing. If you microdose ibogaine, your ibogaine is a prodrug to noribogaine. And keep in mind, lamberine, low-dose ibogaine tablets, were sold in French pharmacies for over 20 years. So they did that experiment. They did the low-dose noribogaine experiment for us way back when.
But they did see some toxic effects.
No.
No, they didn't pull the drug because of this?
No. It got scheduled when all the psychedelics got scheduled.
Oh, I mean in France.
Yeah. When all of that, when the harmonization between what was going on with psychedelics, France has always been very strict about psychedelics, unlike other, the Netherlands or Germany or whatever, where it's not scheduled. But ibogaine is scheduled today in the Netherlands. Excuse me, scheduled today in France.
In France, okay.
But no, it got a bad rep as being a doping substance, because it's a performance-enhancing drug in low doses. But think about ethnobotany, ethnopharmacology. What was used in Africa to help hunters go out at night, not to be hungry, and to stalk their prey. They had stamina. And mountain climbers reportedly were using it. So it got caught up in that hole by the Olympic Committee, and got listed as a potential doping substance. So that's really what took it out of use. And then don't forget too, originally a lot of the pharmaceutical development in Europe before the fancy chemistry out of Germany and Switzerland was what? Mother Nature, right? So, our drugs came from natural products. And the French group had ibogaine for a very long time. That's Professor Gutturel. And he's really the father of ibogaine in terms of the scientific development. So that was all pioneered for a very long time in France. And it becomes, it comes back into the United States, the way I understand it, at the time of psychedelics, when, LSD and there was this underground network of psychedelic therapists out in the West Coast. Sounds familiar today, doesn't it? But this is back a long time ago. And there was the secret chief, who I had no idea who this secret chief was. I must say I wasn't really, I was like, okay, this is pharmacology. We're going to study this, but there's all this mythology even today around, iboga and ibogaine as being, this master plant teacher and all this stuff. But I wasn't in that crowd. I was like this, lone horse with a team, developing a pharmaceutical that desperately needed to get into patients. And keep in mind too, where was I? I was in Miami. Miami was front end hit by the cocaine epidemic because of our proximity to the, Central and South America and the transshipment of cocaine through the Bahamian corridor, through Haiti, comes right into Miami.
And
So
that led you to use ibogaine. The ibogaine you first used it.
That's
Yeah.
what did it. That's what got. I was studying Alzheimer's. I was working on Alzheimer's. I was in the cholinergic nervous system, and I had done my my work. My dissertation project was published in Science Magazine a bazillion years ago, and I was an Alzheimer scientist, at the time, and I had I was really blessed by having the opportunity to do my fellowship at Harvard Medical School in a premier neurology department with Professor Messelem in the department of Norm Geschwin, like a legendary neurologist, behavioral neurologist. So I was very lucky to train with them, and I had already made my mark in the field, and I had my first grants for continuing that work. And all of a sudden, Miami, cocaine, I decided to write a grant into the addiction field, which I really had never published even one paper, and it gets funded on a first submission. And then we get the discovery of cocaethylene, the ethyl homolog of cocaine that's formed when you use coke and booze, and we become the Miami Vice metabolite. So we get, it's just a synchronicity, right? Has its own, I don't know. I'm a scientist. What can I say?
Yeah, fascinating.
It's funny. It's funny how your life takes you in a direction.
So could you give an idea of how ibogaine and noribogaine work, what each molecule does in the brain?
Yes.
And which targets matter most for addiction?
Yeah, that's a great question. That's a really great. Obviously, that's a fundamental question, right? Is the on-target, off-target, and mechanism of action of both molecules, and how much of the beneficial effect is tied to the therapeutic effect is tied to the ibogaine, and what is the contribution of noribogaine? And that has been part of my work for many years, trying to answer that question. And of course, other scientists are contributing also. What do we think today? We know that the oniric effects, the oniric hallucinogenic effects of ibogaine, which is the first part of the ibogaine journey in a patient. So when you give an oral dose of ibogaine to a patient will start to experience that. Some of them describe a hum, a buzz that they hear. And then when that starts, it immediately then shifts to, some of them liken it to a movie or a waking dream state, like a lucid dream. And some say that they're getting like a video, like snapshots of their life review. And that's a very intense phase. And it is tied to metabolizer status. We know this. So the length, you can set your watch, the length of the ibogaine visualization period is tied to whether you're fast, slow, or intermediate metabolizer. And that we knew from the very first 6 patients that were treated in St. Kitts. We saw that because we had the genotype and the luck of the draw. We had a poor metabolizer, someone who was cytochrome P452D6 deficient. And then we had fast metabolizer, who was probably, we didn't characterize it this way, but I believe she was an ultra rapid metabolizer who was an opioid user. She had virtually no visions at all, few, but complete blockade of opioid withdrawals. So we had the spectrum right there in the first 6. And we knew that as soon as it converted over to noribogaine, and the noribogaine reached the Cmax in the blood, the maximum concentration in blood, and noribogaine stays around a lot longer. Ibogaine is cleared from the blood quickly. Half-life about 5 hours, 5 to 6 half-lives, the drug is out by the next morning. So the noribogaine is still elevated in the blood, and its half-life is maybe 5 to 7 days to get full clearance when you take an ibogaine dose. And interestingly, if you take what they call in the underground the flood dose of ibogaine, a high dose of ibogaine, it's likely that ibogaine partitions in the body, because I was able to measure noribogaine and not ibogaine, noribogaine in the urine at over 3 weeks. Okay, so that was very interesting. Back to the target. So the visionary, do we need the trip to reach the destination? Okay, fundamental question. How much of the beneficial effects of the ibogaine are tied to the intense oniric effects of ibogaine? I don't think we know. I don't think we know. We know that patients report that they feel it's beneficial, that the vision, the visions are somehow healing for them, that they get insights through their bad behavior, that they make peace with God, their family. They complete, I like this one, they complete a moral inventory. These are people who have been abusing hard drugs, maybe different for PTSD patients. So the phenomenology of the drug experience and the patient's endorsement of the benefits of ibogaine are clear. Meanwhile, noribogaine. So when we were in St. Kitts, it was fun for me to watch this because I wasn't involved. After they came out of the clinic, my interaction with the people in the treatment was limited because we had therapists and psychiatrists there. So I was more with the medical team and the team in the clinic. When they go into their residence and they were doing their post-ibogaine integration, that I left to the experts. So those were our therapeutic team. But it was fun to watch the patients because these were people some of them were at death's door. Death's door. They had failed the standard of care. They were going to give up. They were like, check please. I don't want to live like this anymore.
Opioid addiction.
Yeah, they were exhausted and neurotoxic. You slam back a lot of cocaine or dope, your brain's you're not thinking straight, right?
Mm-hmm, mm-hmm.
And here they were, days later, they weren't craving. They didn't jump the fence to go score drugs. And you're there with and then the group dynamic was spectacular. Mm-hmm. I mean, they were bonded by this, their journey with the ibogaine. So you had people coming off opiates, people coming that were abusing alcohol, people that were polydrug dependent, people that were, cocaine abusers. You had these different, clusters of people, but they were all kind of joined together now, and they felt good. And they would ask me, what's in me today? Because I feel really good. Mm-hmm. What was in them today was noribogaine. So the pharmacology of noribogaine, it's what we believe from our in vitro, in vivo work is that it's an antidepressant hitting the serotonin reuptake inhibitor, high affinity. It's a partial biased kappa agonist. It's not recruiting beta-arrestin, and it's interesting in that site because kappa is dysphoria, right?
Right.
So mu is to euphoria like kappa is to dysphoria. It's kind of a yin yang on the opioid circuitry. And kappa regulates the neurotransmitter dopamine. So we know that, for example, like when people come off of cocaine and you look at their brains after death, the kappa circuitry is lit up like a Christmas tree. Kappa receptors, boom. So if you've got a partial agonist, you can kind of think of a thermostat. You can kind of dial down on kappa-mediated dysphoria. I think this is part of the reason why trying to wean off of buprenorphine is so difficult, is that you go down low, 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 but then you've still got the kappa rebound. So it makes it difficult for patients to detoxify off of long-term Suboxone.
But that doesn't explain the hallucinations, though.
No, noribogaine is not a noniric. We know this for a fact because we know that when noribogaine is high in the blood in very high concentrations, there are visions.
Okay, so noribogaine is mostly, most affinity is the serotonin transporter. And
there's 4 targets that I think about. And serotonin, reuptake, kappa, partial kappa, agonism. Alpha-3 beta-4 neuronal nicotinic receptor, which puts the brakes on dopamine-mediated reinforcement. And this comes from seminal work by Stan Glick and his colleagues in Albany working with 18-methoxycoronardine. They were the ones that really put that on the map. And we know noribogaine hits that site. We've done that in the DemeRx labs, and we've done that with our receptor screens. But the other one that I'm very interested in is, which I think is fundamental here, is the glial-derived neurotrophic factor. So GDNF bumps up dopamine neurons.
RET, the RET receptor?
Yeah, so it's an important growth factor for dopamine, just like BDNF is for the serotonin neurons. So most of the serotonergic psychedelics, will augment BDNF, but the azepinoindoles, ibogaine, noribogaine, and specifically from the, again, seminal work, brilliant work from Dr. Olson, David Olson, on who has published that, noribogaine is the neuroplastogen, and then the fundamental work again from Dorit Ron's lab in California, at the Gallo Institute, where she talks about GDNF and alcohol use disorder, and it's framed in my brain in trying to think about this class of compounds. So I'm a loyalist to them. They really put these ideas on the map. It's their work, not mine. If you think about why do people get addicted, all right? It's an acquired disease. If you never drink or drug, you're not going to get addicted. Simple. So what happens? First of all, why do people go from recreational use to abuse to dependence? Why? Okay, fundamental question. Some patients we know are comorbid for depression and anxiety, social phobias, traumatic brain injury. So there's a lot of vectors that kind of can get you off the road and get you on the path to addiction. Those are comorbid factors, and certainly depression, comorbidity of depression, we saw very high overlap, and that's well known in the medical and scientific literature. But think about this. So when you talk to patients about why do you want to use crack? I mean, I studied, I consider myself a cocainologist because I did a lot of work in the field of cocaine abuse and studied the brain of people who came to death who had abused cocaine. So I spent a lot of time thinking about cocaine. And I've actually pulled people out of crack houses. That's another story for another day. So and it was interesting because I wanted to see what it looks like. And it looked bad. I was like, why would you do this? Why would you do this to yourself? Because it's such a stereotyped behavior, and the things people do in that setting are just whoa. They're way out there in the sense that the frontal lobe is not connected. You just disconnected your frontal lobe, but the patients tell us It's like a cerebral orgasm, Dr. Nash. Cerebral orgasm. Okay. So they're chasing a memory of a high. It's like that first vodka martini. And you feel good, you got the stimulant effect, and you remember where you drank it. I was here looking at the water. I drank that martini. I felt really good, the clarity. The nicotine that makes you feel good. I'm relaxed. I'm cognitive, awake. You remember that. And so you go back to it. You like it. Part of it is that you probably have a underlying dopamine deficiency syndrome. And again, this is other, seminal work in the field of characterizing the dopamine circuitry in the brain. I worked a little bit on that too, myself in my lab. But you can see where that comes into play. I know myself, I can't take opiates. I hate them. They're dysphoric for me. They are not euphoric. All right? I get sick on them. Probably because I have too much dopamine to begin with. I jump up in the morning and I can grab rewards from everywhere.
And that's the thing, yeah. The only source of reward becomes the drug. And
yeah, you get hijacked, and this becomes your whole raison d'etre to go find dope.
It's also a reaction to stress, potentially.
Well, yeah, sure it is. And I always say, opiates are a poor man's, alcohol's a poor man's opiate, because a lot of that is, relief. And again, people who, would shoot up heroin. And I said, and I met, very lovely, well-educated, they're all people, all walks of life find themselves drunk or on drugs, right? It goes across. There's no group that's not affected, right? But you see a very smart, educated person who has socioeconomic safety nets all around, and they're taking a needle, putting a drug in it, and sticking it in their vein. Like, is this a good idea? What are you thinking when you do this? Again, hanging around and hanging with and going through the journey with the patients was informative to me, thinking about it scientifically. But they would tell me, they said, it's incredible. It's the velvet glove. The velvet glove. So we have cerebral orgasm, velvet drug. Yes, a lot of it is the pain, whatever pain, cerebral pain, and sometimes even peripheral pain, but cerebral pain is a vector to addiction, right? And it's maladaptive behavior. So I like to say, how do you break the cycle of addiction? It's an acquired illness. How do you get out? Do you white knuckle sobriety? You detoxify yourself and then white knuckle sobriety? That's really hard to do.
Change environment.
Yeah, absolutely. And it's cruel, right? And so you go to rehab or you go to detox and you get spun dry, and then you're back out craving and you don't feel good, or you break up with your husband, or something bad happens, somebody dies in your family, and you've got no safety net. All that pain comes back. You can't handle it. You're anxious. You're not sleeping. You're hurting. And you're just, what are you going to do? You're going to reach for the drugs or the alcohol. So I think the noribogaine, why I'm so excited about DemeRx 1001,
yes,
is that the noribogaine, we have to demonstrate this in well-controlled randomized clinical trials, will be that safety net, that through the polypharmacology of the drug, as well as the neuroplasticity, because it is a neuroplastogen, that it can help patients through the early stages of sobriety and detox to get through, to allow the brain to heal and to start to think straight, to develop a better behavioral strategy, So I always,
resist the threats or the stress along the line. Yeah.
yes, pre-contemplative, contemplative, ready for change.
So
And
we'll.
neurons that wire together fire together. So if you get those synapses going and you can re-engage the frontal lobe, because I think of addiction as a disconnection syndrome. You're limbic, you're down here. You need to be in the frontal lobe, because the frontal lobe is what is the executive. You think of the brain as the orchestra, that's the conductor.
And also balance serotonin, dopamine, where.
That would be nice to get dial up on your monoamines.
Yes, and serotonin, dopamine being more on the impulsive reward oriented, and serotonin being more on the projection and the delayed reward. So maybe there's an imbalance there too. But tell me about DemeRx 1001. Is it a one-time reset like ibogaine, or is it a monthly maintenance?
No. It's going to be a daily oral dose. Pill, patch, or depot. We'll learn more as we get into the patients, how to formulate it for different indications. So noribogaine's a pipeline in a drug, right? And once we get the chronic dosing, we completed the multiple ascending dose trial, and that was a lot of learning there, and excellent team that we worked with again. Fortunate. Learned a lot of cardiology again with noribogaine, and we're excited. We're going to, in our discussion with the FDA and the authorization safe to proceed with an alcohol noribogaine interaction trial, is sort of for me, the last piece of the phase 1 puzzle before we embark fully on what I will hope to be a very robust phase 2 clinical trial. I want to see these data come forward in patients. They're waiting for us.
But everyone is talking about ibogaine for PTSD through the executive order. You filed for alcohol use disorder with noribogaine. So does the PTSD momentum help or complicate your story?
I don't see, I don't know, maybe, I'm just magical thinking, but I don't think so. I don't see noribogaine competing with ibogaine. There's a place for both molecules. Ibogaine has regulatory hurdles. We know what they are. Ibogaine has to be administered under full medical monitor. Ibogaine is going to have a very, can only imagine what the REMS is going to look like for this drug. The risk mitigation is significant. Not everybody's going to be able to be a candidate for ibogaine. But they don't compete. And here's, when we were in St. Kitts, and I had the team around, and I had wonderful psychiatrists, Dr. Frank Irvin, who's since passed away, and we just had such a good team down there. And everybody, it was great because we had a lot of visitors come into St. Kitts, so we had a lot of scientists and clinicians and therapists and people that would come in during the treatment rounds. They had a lot of help. But I've never taken ibogaine, so I've always maintained my, scientific one-arm length away from. I would never do it because I wouldn't want to be impacted by that. I want to keep some kind of objectivity here. Okay. My objectivity gets blurred because I feel the pain of patients. That's my emotional, but my intellectual, I can look at the data. If this drug works, it works. If it doesn't work, it doesn't work, and we're going to name it. If it's safe, it's safe. If it's not safe, we're going to name it. Okay? And that's what we have to do, and that's my role, and that's why I haven't taken the ibogaine. And I know a lot of people, I think don't like me for it, but too bad, I don't care. I don't care. But I think that, ibogaine, there's a lot more to learn about that drug. So given the regulatory hurdles, and now we have, this legacy action of President Trump, let's get the ibogaine into clinical trials, and let's demonstrate. Is it is it a better molecule for PTSD than some of the others that are in the pipeline?
MDMA, psilocybin, yeah.
Right, you can ask that question. You know, I haven't worked in that space. Many of the people who came through St. Kitts, we clinically phenotyped them, and they would light up the scoreboard for trauma and PTSD. We did that clinical screening. That was part of our psych phenotype that we did. We did extensive psych interviews at St. Kitts, but we were focused on an addiction drug, right? So I think we've learned what the work of Nolan Williams, doing pre and post evaluations of our veterans. I mean,
Stanford,
it's the Stanford study is one of the most important studies that gifted to the field of ibogaine research. There's no doubt about it. And when we lost him, I was in a deep, dark despair because, he was the first in a long time. He was bold. I mean, considering his stature in the field and the contribution that he gave as a neurologist for depression, and I mean, the man absolutely a legendary, gifted human being, and we lose him to ibogaine. But others are, of course, trying to carry on his seminal work, and that's being done also at Sanera under the leadership of that team. And the benefactor, Sergey Brin. So, I'm hopeful that and by the way, I love our veterans. I'm like, I'm right there. Sign me up. I want our veterans to have access to meds. But again, why should our veterans have to go into the vast underground experiment in all these pop-up clinics? And that's what I fear, is that a lot of patients, families who are desperate, desperate people will do desperate things. So I'm grateful that, Texas and the Americans for Ibogaine and Sanera and others are leading the charge on the ibogaine.
And different PTSD profile can require different psychedelics treatments.
100%.
Well. Yeah.
And it may not be a one and done either, right? Because again, it may not be a one and done. But if ibogaine, and again, there's a lot more work to do. There are pharmaceuticals to treat that are not hallucinogens, right? To treat PTSD symptoms. So do they work well? Well, again, you got to do, I don't know because I haven't studied them personally with patients, but, you have to look at what's out there in the literature. I think the other factor, I would say, is people who come to ibogaine for addiction, which they're doing that, some of them are microdosing ibogaine a month later when they go home. That suggests what we already know, that we can keep the window of neuroplasticity open longer for patients who have elected to take ibogaine by giving them a noribogaine as a pharmaceutical, as DemeRx 1001. So to have the noribogaine available to patients as a follow-on medication is a safety net, especially for opioid patients who want to detox ibogaine, and then they come back and they have nothing, no safety net. What is their choice? If they don't want to go back onto buprenorphine, to go back onto a chemical dependency on buprenorphine, what are their options? They don't have many. Naltrexone, not everybody tolerates naltrexone, so let's put noribogaine out there. Let's test noribogaine for OUD and see if patients who have detoxified either with the standard of care, perhaps with a non-opioid detox like lofexidine, clonidine, and benzodiazepine stabilization, come off of it. They're in the earlier stages of recovery. Some are going into rehab, wherever they're going, outpatient. Let's put noribogaine into the formulary and get that into patients.
Did you get a correlation in therapeutic benefits with the genotyping of the cytochrome?
We did not stratify. Too few. Because remember, everybody's got noribogaine on board. Even the poor metabolizer, 10% goes through the lesser cytochrome. All right. So it's kind of like a depot. Because if you're not converting the ibogaine, it's going to go into fat, so it's going to depot in the body, and then it'll come around and leach out and go through the liver and form noribogaine. So you might have a much longer half-life. Again, we were limited in what we could do in St. Kitts, and we didn't, have we don't know, and we saw the graphs, and they were like this, flat. Okay. So that noribogaine. It's a good question, but the answer is no. We didn't have and I wouldn't do it that way. I think you're much better off genotyping the patients, and you group them into intermediate and fast metabolizers. Poor metabolizers fall in a separate box, and then look at your blood levels. And I without disclosing more proprietary information, I won't say more.
Other groups are building analogs that strip the psychoactive and cardiac effects. Do we need a new molecule, or is noribogaine the answer?
Well,
I'm asking the right person.
Yeah, you're at. Well, I really want a medicine to get available to people. I obviously am advancing noribogaine, and we have de-risked that molecule, right? Because it's been in patients. Everybody who takes ibogaine has noribogaine. So we've already got somewhat of a de-risk here. I would say we have a big de-risk, all right? Because, you can put drugs into animals, and we've had a lot of drugs in animals that have never made it into people. The screening, for drugs of addiction, this drug stops drug taking, this drug stops drug taking, this drug stops, and then you bring it into people and it doesn't work. Also, animal tox. You don't know that you've got a drug that's going to go into people until you do the toxicology, right? And sometimes, a new molecule is going to have off-target effects that you couldn't predict, based on structural similarity to the lead compounds, either ibogaine or noribogaine. You take them as lead molecules. So short answer is noribogaine is a de-risked molecule. It's got a robust pharmacology. We know it's a neuroplastogen, and these are published studies, not by me, by other people in the field. Can we dose it safely? DemeRx has demonstrated, yes, and the FDA has authorized the next study. Now we're really positioned for a phase 2 clinical trial. So once we do that interaction study, we're there. We're right at the gate. Of course, we have to raise dollars, but we're working on that too. So I'm optimistic that we get the drug into patients quickly, and we're ahead of the curve of the other molecules that are coming. Now, who's to say that these molecules aren't going to be better in some way? They very well may be. They very well may be. Or they may have even more novel indications. Maybe they're better for something else, that we didn't anticipate. Are they going to knock noribogaine out, off the train track? That's what science is about, right? The bottom line is, I really pray for all drug developers in this space, because people suffer. Our country needs it. It's a global problem. And if this class of molecules, if the azapinoindoles, if the ibogaine-like molecules, any and all, whether it's the tabernanthalog series with DemeRx, or the Gilgamesh compounds, which it seems like they're betting on noribogaine analogs being the winner, or our work, it's all good. It's all good.
One last question for you. Do you see the right to try access for DemeRx 1001 like next year, for example? Or where do you see DemeRx 1001 next, like say, 5 years?
I mean, that's going to be, I That's a very interesting question. All right, that's a really interesting question. So it's too bad that I think right to try is going to be more important for ibogaine's clinical development. And there's a lot of, it sounds like a great idea on paper, and then you get into it and maybe it's not such a great idea, right? Because you let the genie out of the bottle and then people make mistakes and then you got to own the mistakes. So that's why companies don't always allow patients to try, right? And there's more to be discussed about that. We don't have time. For noribogaine, we don't have the blinding issue that ibogaine has. So we're going to be able to hopefully quickly demonstrate a robust and lasting, enduring efficacy efficacy marker for this drug. If we can demonstrate that, I think the FDA will work with us and help us accelerate, clinical development to a point where we have a label. We need the efficacy signal. We have to be able to show this. For ibogaine, it's going to be different. For ibogaine, it's going to be benefits outweigh the risk. That's a high bar. Noribogaine risks, not so much. We don't have that. We got to show that we've got target engagement and that this works well. So you see what I'm saying? So I think the right to try, and that was discussed in the executive order, is something that could give us a safety database for ibogaine. And I would like to see that happen. When I was running ibogaine studies more recently in our company, I talked about it. I told our board I wanted to see it happen, and DemeRx would have been very willing to sponsor it with the guidance from the FDA and the REMS. Whatever the risk mitigation, we'd have to be very strict about it. But I don't want people to have to go offshore. I don't want people to go outside ex-US for treatment.
Yeah. Well, thank you so much, Dr. Mash, for your insights, and congratulations again on this historic moment. Stay with me 1 second, and thank you everyone for tuning in. I hope you enjoyed our conversation. Feel free to message me if you have any questions that I can forward to Dr. Mash. Thank you so much, and see you next time for another episode of Cascades Analytics Live. Thank you.